Perfect. So welcome to today's Creekfest 2020 webinar, all about whirling disease. My name's Robin and I work at Bow Habitat Station in Calgary. So I just wanted to kick off our session by respectfully acknowledging that I am joining today from Treaty 7 territory. And I'd like to acknowledge the many First Nations and Métis whose footsteps and gatherings we have marked this land for centuries. We honor and respect the elders, past, present, and future. And I just want everybody to take a moment to acknowledge the Indigenous people of the regions that you find yourselves in today from wherever you are joining from. So for those of you who haven't been down to Bow Habitat Station, it's kind of like a science center, but it's all focused on nature here in Alberta. So visitors that come can learn about Alberta's ecosystems, air, land, water, wildlife, and fish. And we are very lucky here that we're located right next door to the Sam Livingston Fish Hatchery, where over 1 million trout species are raised every year as part of the provincial stocking program. So what that means is in the spring, they actually get released into lakes and ponds across the province. There's about 220 of them. And so because they are going out into the wild and interacting with wild ecosystems, we have a lot of work to do to keep those fish healthy and keep the facility free of any excess bacteria or diseases, such as whirling disease, which is our hot topic today. So I don't know if you've heard about whirling disease before, but if not, back in 2016, whirling disease popped up in Alberta here, and it did pose a risk not only to our wild fish, but also the fish in the Sam Livingston Fish Hatchery, as well as other provincial fish rearing facilities in Alberta here. So we are very lucky at our facility that we are whirling disease free. We actually treat our water with UV light to destroy any bacteria and also to destroy the spores, which we'll hear more about later in this morning's program. But today we want to know what whirling disease is and learn more about it and what it impacts and kind of where it is in Alberta as well. So we are so lucky to have biologist Clayton James on the line with us. Clayton, can you give us a wave there?
Hey, everybody.
Perfect. And we will pass it over to Clayton very shortly here. He's the subject matter expert and has a lot of knowledge to share with us this morning. But before we do, I just wanted to give everybody a bit of a crash course in Zoom and some of the functions that we'll be using today. So along that black bar at either the bottom or the top of your screen, it should look something like the one up on screen right now. And we are going to be using primarily the polls function. So what that is it's a quiz function, and we're actually going to practice by answering a poll right now. So I do encourage you to participate. I will let you know that all of these polls are anonymous, so don't feel like there's a wrong answer. We will discuss the answers and how some of them are, well, there are some wrong answers, but not in a way that will reflect on anybody at all. So I'm going to launch this first poll here just as a practice, and you should see a dialog box popping up on your screen right now that says, had you heard of whirling disease before today's webinar? Excellent. I see some of you are getting the hang of these polls already today. I'll give people about 10 more seconds to answer. And while they are doing that, I'll just let you know that this dialog box will stay on your screen, so you're welcome to close it between polls. Every time we open a new quiz, a new dialog box will pop up on your screen. Otherwise, it kind of is just in the way and you won't be able to see everything that Clayton is sharing this morning. Okay, great. So we all answered, and I'll just share our results here. So most of us have heard about whirling disease before. That's excellent. There's lots more to learn from Clayton this morning, and a couple of us haven't. So hopefully we're all on the same page by the time we end today's webinar. Excellent. So again, if you wanted to, you can close that poll box at this time. And then the other functions that we'll be using are either the Q&A or the chat function. Both of those are great places that you can ask your questions as we go today. And I might interrupt Clayton along the way if some of the questions are relevant to what he has on screen at the moment. Otherwise, we'll address some of those at the end today. But I will let you know that while you can see and hear us, we can't actually see or hear you. So using that Q&A and chat function are the best ways to go there. So with that, I will pass it over to Clayton. So Clayton, before you get into the meat and bones of your whirling disease presentation today, can you tell us a bit about your job and what you do day to day?
Yeah, absolutely. Thanks a lot, Robin. I really appreciate the introduction there, and it's good to see so many folks participating today to learn about whirling disease. I'll give you a little bit of actually my background. I grew up west of Edmonton near Stony Plain. I spent most of my life there, and I grew up really enjoying fishing in the outdoors. And that led to me pursuing a university degree in biology, and eventually I got a master's doing fish parasitology. So I've been working as a biologist, a fish biologist, for over 10 years now, and most recently, as of about 3 years ago, I accepted a position with the government of Alberta to work on whirling disease. And this is a very big interest of mine, as I've studied fish parasitology during my studies. So day to day, there's a lot of things that a biologist does. We spend our summers mostly outside trying to examine the things that we're studying, such as whirling disease and fish in particular. And in the wintertime, we like to take all of that data that we collect throughout the summer, and then we like to look at it and see if there's any kind of trends that we should be concerned about, or that might be very interesting. And then we try to put all that together in a report and distribute it to the public or interested people that want to learn more about the subject at hand. So that's what we do day to day. So I think I'm just going to move onto the presentation here. And again, as Robin said, what I'm going to talk about today is mostly about what is whirling disease. But in particular, I want to learn about, or I want to tell everybody about sort of what it means to Alberta. So first, when we talk about whirling disease is a disease of fish, and it's caused by a microscopic parasite. And this parasite has been around the world for a very long time, longer than pretty much anybody knows. It actually originated somewhere in Europe, and we know for sure that Germany is where we believe the parasite traveled from and came over to North America into the United States. In particular, the United States in the mid-90s saw some major effects to fish. So it's a parasite that affects fish. And when it is in a high enough infection, it causes whirling disease. So there's a very important distinction there. There's the parasite that causes whirling disease, and then there's whirling disease. Only recently, though, we discovered that the parasite has come to Alberta. And that happened we discovered it in 2016 in Banff National Park. So that's very important. And just to highlight that, you can see it in 2016. And so it's been, we know for sure it's been around for about 4 years, but we're not sure how much longer it's been around before that. I'm going to kind of switch a little bit to focus on a topic that's a little bit more relevant to today, and that is the coronavirus. So we're all dealing with COVID-19 in our day-to-day life, and as many of you probably know, it's affecting the way you do things, how you learn for school, etc. So I wanted to kind of compare what whirling disease is to fish and how that is different from what coronavirus is to humans. So first of all, COVID-19 stands for coronavirus disease. So it's very similar to whirling disease in that it has an impact on its host. And in the case for COVID, we know that the host is humans. For whirling disease, the host is fish. So that's a similarity. But there is actually quite a bit of a difference as well. And one of the main differences is that coronavirus is a virus, and a virus actually isn't alive. Whereas whirling disease is actually from a parasite that is a living organism, a microscopic living organism. And it's very closely related to jellyfish, which is it's very interesting, and not very many people know that. And another distinct difference about COVID and whirling disease is that COVID, a human can pass it on to another human. Whereas whirling disease, a fish can't actually pass it on to another fish. It actually goes through another organism, which is a little worm species, which I'll explain a lot more in more detail. And it's the worm that actually releases the parasite in the water that infects the fish. And one major difference because of that life cycle and the difference between humans and fish is that fish, they don't know they're getting infected. So they can't take the same measures that humans can take. So you've probably seen that a lot of people are wearing masks to help prevent the spread of COVID. Well, fish, as I show in the picture up on the top left there, fish can't wear masks. So they don't have the same resources. So they kind of have to rely on their own immune system in order to prevent, or in order to fight the disease. Okay, so based on what I just informed you, I'm going to do a little quiz here. And so this quiz is what can be infected by whirling disease. Robin, are you going to take over from here? Okay.
Yeah, there we go. Sorry about that.
Yeah, no worries.
We'll give people about 10 more seconds to vote. It looks like lots of people have quite a bit of knowledge about whirling disease already. This is great.
Yeah, it's awesome.
All right, and let's see what people said here. So this was a multiple answer question. So it looks like everybody did select fish, and some folks thought that whirling disease could also affect wildlife, people, and pets there.
Okay. Awesome. So just to go through an answer here, if we focus on wildlife, it actually can't affect wildlife. So no animals that are in the wild. As for people, it can't actually infect people either. And this includes eating a fish that has whirling disease. That won't affect people. Also pets, it doesn't infect pets. Basically, the only species that it does affect are fish. And I have a check mark on the one in the water and the one in the bear's paws there. They're the only ones that are going to see any kind of trouble from this parasite. There is a bit of a caveat though, it's not all fish. So up on the screen here, I have a bunch of different types of fish that are actually common to Alberta. If you've been fishing before with your parents, you might recognize some of these fish. And if you haven't been fishing before, you can trust me that you'll probably be living in close proximity to some of these fish. If you live in Edmonton, for example, you may know of this fish, which is called a walleye. And if you live in Calgary, you may know this fish here, which is a rainbow trout, or this one, it's a brown trout. And if you can recognize them, that's wonderful. That's wonderful. So whirling disease doesn't actually infect all fish. It only infects members of the trout and whitefish family of fish. So we're going to jump into another quiz here, a fish ID quiz. And I don't know if you can see, but there should be letters above some of the fish there, listed A, B, C, and D. So can you tell me which one you believe out of those four is a member of the trout and whitefish family?
And while people are answering, we have had some comments saying that people recognize some of these fish, so that is great. Clayton, while we're waiting, we have a question coming in as well. I've heard some aquarium fish can get whirling disease. Can you confirm or deny this?
I would say that unless your aquarium fish is a member of the trout family, so any kind of trout or whitefish, then probably not. So you wouldn't be able to have those as aquarium fish because you require a special license. So no aquarium fish that you would buy at a pet store would be able to get whirling disease.
Great, thank you.
You bet.
Okay, so let's see what people think about which fish are part of the trout and whitefish family. So we have quite a few votes for A, B, C, and D as not being part of the family here. So Clayton, which ones are part of the trout and whitefish family?
Yeah, that's great. So it's quite a mixed result there. So the answer to the question is actually D. D is what we call a bull trout and is native to Alberta. So in addition to a bull trout, in this picture here, I'm going to go through a few other members of the trout and whitefish family. Up on the top right, you'll see one of the whitefish, and this is called a mountain whitefish. You find this a lot in rivers and streams near the mountains. The next one up on the top left over here, this is called an Arctic grayling. It's not known as a trout or whitefish, but it is a part of the trout and whitefish family. And then below that in the bottom left, that's a brown trout. And then here in where I'm circling with my mouse is a brook trout. And then over here on the right, it's a cutthroat trout. And down at the bottom in the middle is a rainbow trout. This is probably, these three trout over here, the brook trout, cutthroat trout, and rainbow trout are probably some of the more common trout you're gonna find in Alberta. Now the other fish that were listed there, that includes a walleye, which if you've been fishing before, you probably have seen a walleye if you've been fishing with your parents. And this is a yellow perch, which is a smaller fish quite closely related to walleye. And then this one right here is a northern pike. And the one on the far left is a goldeye. You might find these actually in the North Saskatchewan River. These fish are not part of the trout and whitefish family, so they can't actually get infected by the parasite. And interestingly, all of these other fish that are part of the trout and whitefish family, they can get infected, but not all of them are going to get whirling disease. Some of them are just more resistant. Their immune systems are stronger. And as a result, there's actually only 4 species in Alberta that we're really concerned about when it comes to whirling disease. And that's the mountain whitefish, the cutthroat trout, rainbow trout, and brook trout. So for those, I noticed there was a comment that they're having success in identifying the fish. If other folks are at all interested in fish identification or just fishing education in general, I'd like to point out that right now, there is an online course that is offered by Alberta Hunter and Education Association about Alberta fishing education. It's currently free. It's generally about $40. So I believe, and maybe Robin, you can confirm with this link will be posted so that folks could access it sometime after the webinar.
Yeah, absolutely. We will post it on Bow Habitat Station's Facebook page just after this webinar.
When I was in elementary school, I actually took this course as well. It's a very good course, and it offers a lot of what you've just seen here. Okay, so how does the parasite that causes whirling disease infect fish? Well, I have up on the screen here just an image for illustrative purposes. So I need you to keep in mind that you're not actually going to be able to see the parasite that causes whirling disease. They're not going to be big like this so that when you're walking in the river, you can see them. But this is just to highlight what it looks like from that perspective, from the worm and from the fish host. And so the parasite, as I mentioned, is closely related to jellyfish, which is really weird because it actually requires different life cycle stages to be able to get to the fish. And the first stage I'm going to talk about, it's called a myxospore. And so this myxospore, it sits in the bottom of the river or the lake. It doesn't do anything, it doesn't move, it just sits there. And then inadvertently, a worm known as a tubifex worm may come by and accidentally eat one of these or a couple of these myxospores, and then it will become infected. The worm doesn't know if it's eating it, it just will accidentally eat this spore. And then after a period of time, when the worm's infected, it's going to release a totally different spore stage of the parasite, which is called a triactinomyxon, which I know is a bit of a mouthful. And so we call it just a TAM for short. And a TAM is actually a very fragile life cycle stage. Kind of looks like a grappling hook, and it's neutrally buoyant, which means it kind of just goes with the flow of the water. And these little guys here are what's going to infect the fish. So the moment they come in contact with the fish, they're actually going to penetrate through the skin and go to the site where they want to infect. Once the fish has been infected after a period of time, it's going to create these myxospores again. So when the fish dies and it settles out at the bottom of the lake or the river, it's going to release these back into the environment, and then more worms are likely going to consume them and start the life cycle again. So another interesting comparison between COVID and whirling disease that I'd like to point out is that for COVID, we know that the most vulnerable people in society are generally older people. But for whirling disease, the most vulnerable fish in a fish population are actually the youngest fish. And there's a reason for that. The parasite that causes whirling disease, it feeds on cartilage. So not bone. So if you think about your ear, that sort of rigid but softer tissue, or the front of your nose, that's cartilage. And when a fish is first hatched until about 10 weeks of age, before it reaches that 10-week period, its entire skeleton is cartilage. And so if we were to open up a fish, this being a young fish, all of that skeleton is cartilage. So the parasite will go, once it's infected by the TAM, will start feeding on that cartilage. And what happens if there's enough of the parasite and enough damage is done, the skeleton will not develop properly. And when it doesn't develop properly, you start getting some deformities in, like, let's say the spine, for example, or the tail, or sometimes even the skull. And then you start getting these signs that you see in these young fish. They can include a black tail or a bent spine or a very short head. And what that looks like, I'm going to show you now what that looks like in fish from the wild. I actually have two pictures here of fish that I caught myself last year in a river in southern Alberta. And the one on the left here is what I would call a very healthy individual. And the one on the right here is one that we can see just by observation is infected and experiencing whirling disease. And so there are obvious different signs between a healthy and an impacted individual. The first one that jumps out is the black tail. So we see this a lot, actually, and in very severe infections. The black pigment color is actually because there's pressure on the nervous system, which changes the color of the tail. And this can occur throughout the body, but generally is restricted to the tail area. Also, if you look at the healthy fish on the left, you see that the tail's quite straight. But the one on the right, where it's impacted by whirling disease, there's a big bend in it. So that's a spinal deformity. A more subtle sign of whirling disease, actually you can start to see, is the eyes of the fish start to bulge a little bit. And the reason why they bulge is because if you look at the snout of the fish, it's quite a bit longer in the healthy fish than it is in this whirling disease impacted fish. And that's because the parasite is feeding on the cartilage in the skull area, and this causes this sort of bulging of the eye and a shortening of the snout, and sometimes we get what's called a sloping forehead. Another sign that we see generally, actually quite frequently, is the gill plate is reduced. And so the gills are where the fish breathes. And in a healthy individual, this gill plate, it completely covers the gills. But in a whirling disease impacted fish, as you can see the gills very clearly. And that's because, again, the parasite is feeding and deteriorating that cartilage on the gill plate. This poor guy also had a deformed jaw. As you see, this jaw is more outward, and the one on the whirling disease fish is kind of inward. So I'm hoping you guys are paying attention to this, because at the end of the talk, I'm actually going to quiz you on some other fish we've captured in some of our previous work. In the most severe cases, when all of these deformities happen, you can actually get instances where fish, their swimming behavior will be affected. What I want to show you here is a video from YouTube. Oops. I might need some help here from Robin, because it's a video and it doesn't seem to be activating.
Yeah, Clayton, I will, if you wanted to move to the next slide, I can get that up here and then we can come back to it if that works.
Yeah, sure. What we're going to show is that the behavior of the fish when their skeleton doesn't develop properly is that they swim in this an interesting behavior called like a whirling pattern, and that's why we actually call it whirling disease. And when they're exhibiting that behavior, it makes it very challenging for them to feed themselves, as well as it's much more challenging to avoid predators or other big fish that want to eat them.
Okay,
so
that should be good to go there.
Okay, great. Thanks, Robin. So if you pay attention, so this fish, before we even start the video, I'd like to point out a couple of things. It's about the same size as those fish that I showed you before. And also you can see that obvious black tail again, and you can see a bit of the bulging eye in the gill present. So it should come swimming here. You see how it does that quick whirling pattern? That's it right there. That's very characteristic of whirling disease. It's a very tight concentric circle. And as you can see, it would make it very challenging for that fish to be able to feed on whatever it's trying to capture, but also would make it challenging for the fish to avoid predation. Yeah, it's not a good scenario when a fish is heavily infected. I see that from Keenan. Okay, so can we move on from that, Robin?
Yeah, of course. Oh.
In those situations, as I mentioned, oh, I don't have that mouse again. Let's.
Sorry, Clayton, 1 second. I will give that back to you.
Yeah, sure. In the situation where a fish is impacted severely enough by whirling disease and it dies, as you see, it's just going to go to the bottom, and all of those parasites that become spores in the fish again are going to be released on the bottom, and we'll start that life cycle all over again. Okay, so well, how do we as biologists study whirling disease, and what exactly are we looking for? I'm going to show you some stuff that we've done last year on a specific river, which is called the Crow's Nest River down in southern Alberta. And first, I'm going to show you whereabouts the Crow's Nest River is located. So as I mentioned, it's quite far in southern Alberta. it has been confirmed positive for the parasite. So this map of Alberta here shows a lot of these red zones, and these red zones are rivers that we've confirmed are positive for the parasite that causes whirling disease. But if you recall, I mentioned there's a very distinct difference between having the parasite and having whirling disease. A fish can have the parasite and never get whirling disease. It's much like COVID, how some people can be completely asymptomatic. But in severe cases where you suspect it's got whirling disease, where the infection is very high, you can start seeing a lot of those signs that I showed you before. So the Crow's Nest River is down here. It's a very popular river for fishing and recreation in southern Alberta. This is another representation of the life cycle that I've shown you before, and it seems like it's a little bit complicated, but I assure you it's not any different than this last life cycle I've shown you. The reason I want to show you is because I want to show step by step what we're looking for as biologists in the field. And if you can see here, there's similarities. So when we talk about that TAM, or the free-floating stage that infects the fish, there's this part of the life cycle corresponds to the TAM that I showed you before. We talk about the fish, same thing, and then the worms eating the mix of spores down below. There's also correlated to the worms eating the mixed spores in this other diagram. The only real difference with this life cycle that I'm showing is that the adult fish don't tend to die by whirling disease. And so they'll die by natural causes, whereas the young fish, which we have represented here on this second arrow, they're the ones that are going to experience the disease the most, like the whirling or the blacktail, and may die as a result of that. So let's start with the worms. This here is what a tubifex worm looks like under a dissecting microscope. You can see that a very obvious coiled pink pattern. And so we want to look at these in the river because we know that these worms are actually what's responsible for infecting the fish. And so how we do that is if you take an area of river, they do burrow. That's a very good point, Keenan. We like to focus on the sort of near the shoreline where there's a lot of mud and organic material, because that's what the worm likes the most. And so we'll go in there with a fine mesh net and we'll kick and look for the worms. So it's a very rigorous kicking, and you're trying to really disturb that mud. And then after that, you can collect here on the, if you can see my mouse, that's actually what the size of a tubifex. So you can see them with the naked eye, but they are quite small. Once we take our mud sample, we will then look in a tray, and we'll look for more of the worms so that we can get a general idea of how many worms are in that river in particular. And so we've spent hours and hours looking through these trays for these worms. And then we try to pull them out with a pipette. So here's an example of what the worm looks like in the pipette. Now I mentioned that the parasite, the floating stage, that TAM, it's microscopic, but we can actually, as biologists, we can look for that in the river as well. So how we do that is we'll go out into the river and we'll take a bunch of buckets of water and we'll pour them through a very, very fine filter. And a bunch of very small things will get caught in that filter. And we take that small stuff back to the lab with us and we'll stain it, and then we'll look under the microscope for that infectious TAM stage that we talked about. And basically we'll go through a slide and count as many TAMs as we find, and we get a general idea of how many parasites are in the water infecting the fish. We can also look for the disease, as mentioned, by looking at small fish. And how we do that is we can do two things. One thing, we can put small fish in a container that has a flow-through screen and monitor them over time. So the parasite is allowed to flow through, and then over time, we can see if those fish develop the disease. In addition, we can actually look at the wild fish, and we do this through a technique called electrofishing. And some of you may have heard of it, some of you may not. And what electrofishing is actually a backpack unit that has a battery, and we put electricity into the water. And that electricity will temporarily stun fish. It won't kill them, but it'll temporarily stun them. And then we have another biologist that's waiting to catch them with a net. And once they catch the stunned fish, we can then look at them and measure them. And we can look for those signs, as I mentioned, of the disease that you've seen before. And then there's the other parts to measure whirling disease, and that's the spore in the fish itself. And so how we do that is we'll take those fish that we collected from our cage, and we can actually send them to some very smart people in the lab, and they will tell us through a variety of tests whether or not those fish have the parasite for whirling disease. So as you can see, biologists, when we are studying whirling disease, there's all sorts of different ways we can study it, and we are quite busy when we're able to access the river. So what did we find, and sort of what are we looking for? So this is an interesting infographic for you. So one of the main findings that we learned from the Crow's Nest River from our study last year was the amount of parasites that we found in the river, and how did that compare to other rivers that have previously seen whirling disease. So on the left here, you see a bunch of rivers, and to be able to visualize how many of the parasite are flowing through the river. You can see that the Crow's Nest River is the second highest in this group of rivers. These are all rivers that were sampled in the same way in Colorado in the early 2000s. And each one of these rivers we know had a very dramatic impact on the fish population. In fact, they saw over 90% decline in rainbow trout populations in all of these other 5 rivers. So the Crow's Nest River is not keeping very good company because it is seeing similar signs and similar numbers of parasite compared to these other rivers. Another interesting result we found with our electrofishing efforts is we're able to get a snapshot of what the population looks like for fish. So a healthy fish population is represented by the top of the screen here. And a healthy population will have fish of all different ages. So ones that are freshly hatched, 1-year-olds, 2-year-olds, and growing all the way up to adults. And should be many fish in each one of these different age groups. In a low severity situation, you have some individuals that are infected. And in this visual here, the ones that are infected are the ones that we see highlighted by the red. But in a low severity situation, you're probably not going to see signs of the disease. And this is kind of that case of the asymptomatic, where fish aren't likely dying from the disease or the parasite. And so you can still have what looks like a very healthy fish population. If we increase to more of a moderate severity, though, this is where we start seeing more fish infected, a higher proportion of those fish in the population. And you'll start seeing signs of that disease in the younger fish. And then there will also be, some of these fish won't grow up to be a year old. And so there won't be as many fish in your fish population. And in a high severity situation, very, very few fish are going to survive, and all the fish will be infected. And so as you can see, if no fish are surviving to one, you start to get this gap in fish. There's no more 1-year-old, 2-year-old, or 3-year-old, etc. And just the adults remain, because those are the fish they're not usually going to die from the disease, but they will die of natural causes. And you can imagine when these fish die, there's just going to be no fish coming behind. And that's how they can see such major declines of 90%. So this is a very serious concern in Alberta. And our work that we did last year is very concerning for the Crow's Nest River, because this other visual below kind of shows you what we found when we look at all those different life cycle stages, the worms and the fish and the parasite. We find that at the top of the watershed, so where the river starts, it doesn't seem to be much affecting, but as we go further and further downstream, the severity of whirling disease is quite high. In fact, at this lower section here, we're seeing almost 100% of fish are exhibiting the disease. So it is a concern for Alberta, and there are things that biologists are working on. We want to find out more if there's more cases of whirling disease occurring in those other rivers that were positive for the parasite. But I'd like to show you some of the pictures of some of the fish we caught at that lower watershed there in the lower Crow's Nest River. So we're actually going to try to test your ability to detect whirling disease in some of the fish that we sampled. So this is the first group. So I'd like folks in their poll to identify which fish they believe have whirling disease. You can select more than one. And we'll give you the freebie for number one, because apparently it's already checked off.
Clayton, while people are voting, we do have a couple of questions. So one of them is back when we were talking about the different species of trout and whitefish that are susceptible to whirling disease. So there was a question saying, or asking, sorry, if those species closely related to rainbow trout, such as golden trout, are also susceptible to this.
That's a really good question. So golden trout are actually quite rare in Alberta. There's not very many populations of them, but they are very closely related to rainbow trout and cutthroat trout. And those, I would say, arguably, rainbow trout and cutthroat trout are the most susceptible to the disease. I don't think golden trout have been tested particularly, like specifically, but I would say that because they're so closely related to rainbow trout, they would be very susceptible to whirling disease.
Perfect. Okay, it looks like most of our participants have voted here, so I'll share the results.
I see there's a question about hybrids. I'll answer that in a second. Yeah, so are the results all in then?
Oh yeah, sorry. I
okay,
assumed you could see them on screen there, but
I can now. Okay. Yes. Just before I go on to the results, there was a follow-up question about hybrids. If there's a hybrid, because rainbow trout and cutthroat trout actually hybridize quite frequently, and particularly actually in the Crow's Nest River. And yes, the hybrids can also be infected if they're the vulnerable species. It may change how affected they become, but I don't think a lot of work has been done on hybrids specifically. So to answer the question, it looked like a lot of you folks were able to pick the ones out that had whirling disease. And in this case, all of these individuals did have whirling disease. And these are all very severe cases of it. As you see, there's that black tail in all of them. It's very obvious. The gill plate is shortened where you can see the gill exposed on some of these. This guy in the top right in particular has a very bulging eye with a short nose compared to the other ones. So yeah, all of these four fish are very infected with whirling disease. So I have another one. It's a little bit trickier. Another 4 pictures for you. So we're going to go back to the poll and see if you can determine which, if any of these fish have what you would suspect would be whirling disease.
it looks like this one is a little bit trickier for folks. So based on what we've talked about, feel free to make some guesses. It's totally fine if you guess incorrectly. Clayton will talk us through which ones have whirling disease and which ones don't. So don't be shy.
I will say that biologists can often get this wrong as well. So this is not, it's not that simple. We are, as mentioned, whirling disease is quite new to Alberta, so we're also learning.
And I will confess that when we went through this presentation yesterday, I would have guessed the wrong one, so. Okay, it looks like most of us have voted, so we'll share the results with folks here.
Okay, so the results, there's a few folks that identified I think each one. So the ones that actually have whirling disease here, if you if you guessed number 7, you were correct. This one is very, very subtle. It is very challenging, and again, when we're working in the river, finding this, sometimes we miss this as well. But it is a sign that we see a lot more frequently, and this is that shortened gill plate. So if you were able to see that the gills are exposed here, the individual 5 here is actually what we would consider a healthy individual. And again, you don't see those gills exposed. There's also a bit of a difference. You can see the nose is a little bit shorter in this case than say a longer one like that. But this is a very, very, it is exhibiting whirling disease, but very lightly. And also, if you pointed out 8, that is also correct. This one had whirling disease. And this one we can tell a little bit more because of that really bulging eyeball and a very, very short nose. So we do tend to see this once in a while where the eyeball becomes very protruded like that. You can see the difference in a lot of these fish. The eye, the skull over top of the eye is very obvious, but not in this one. It's mostly just the eye.
And sorry, folks, if you tried to vote for multiple and were only able to vote for one, that was some technical difficulties on our end, so we apologize for that.
Okay, so I'm going to move on to sort of what you can do about it. For us as biologists, we like to inform ourselves on where this is happening, and then once we know where it's happening, then we can make a more reasoned approach about how to deal with it or how to help solve it. And I will say that the US has had this parasite for a long time, so we're not learning this as we go, and we do have a bunch of friends in the US that I often talk with quite regularly about what to do about challenging whirling disease situations. Usually it all comes back to a very common point, and that common point is that if you have the parasite, it's a lot harder to deal with it than if you don't get the parasite in the first place. And so this is sort of where people like yourselves come in when you go and you recreate in the water. What our major goal is to prevent ever getting the parasite in the first place. Because as I say, once you get it, it's a lot harder to manage. So let's say, for example, you decide you want to go fishing in, let's say, Calgary, which we know Calgary is positive for the parasite. And so you go for a lovely day of fishing in a lake within the city, a stocked lake that has the parasite, and you're tromping through the lake. Well, while you're moving through that lake, anything that touches water has a chance of actually coming in contact with the whirling disease spore, that myxospore that sits on the bottom. And it can attach to all your gear, and it can stay there. And then let's say the next day or even the same day, your family decides we want to go to the mountains and go do some water recreation. And so you think to yourself, I'm going to go fishing in a mountain stream where there wasn't previously the parasite. And so you're about to step into that water with those spores. Do your best, do not do that. That's what we're trying to encourage, because that's how you start the life cycle in an area where it hasn't been before. But this doesn't just apply to people who are fishing. This can apply to all forms of water recreation, including kayaking or swimming. It doesn't just apply to fishing. So anything that really touches water can transfer the parasite. And so what we mainly try to emphasize as biologists is to clean, drain, and dry your gear. So once you get out of any water body, make sure you take the time and effort to clean all of the lake material that is accumulated onto your gear, and clean it and rinse it really, really good, and then let it dry for at least a day, and preferably, if you can, in sunlight. And then your gear will be better protected and not carrying that parasite to transfer. So there's a quiz here. Robin, I'm going to let you step in here because I didn't quite is the quiz to test what kind of gear can be contaminated?
It is, yeah. So based on what we just learned from Clayton about cleaning, draining, and drying our gear, this is just a quick question about what gear you should clean, drain, and dry after each trip out to a lake or river, either here in Alberta or elsewhere if you're off traveling somewhere. So this one you should, again, be able to select multiple answers, and if there's a glitch, I apologize for that. Excellent. We'll give people about 10 more seconds here. All right, so it does look like there is an issue with voting for multiple things. So I will just assume that the things that have been voted for, there's some consensus on those ones. So we'll share these here. Clayton, did you want to talk us through these answers?
Yeah, sure. It looks like most folks got it exactly right. Anything, so we had boots, mobile phone, fishing rod, paddle, and life jacket listed. The only thing that wouldn't contact the water would be your mobile phone. You wouldn't want that to contact water, so you wouldn't have to clean that for whirling disease. But yeah, if your boots or your fishing rod or your paddle or your life jacket come in contact with the water, it's the best case to clean that gear before you go to any other water body. Okay, good job, everybody. So now that all the folks attending, now that you sort of know what to look for, if there's ever a time when you are by a river that might have these fish species, these are all the different members of the trout family we sort of talked about. And now that you know what you're armed with, what to look for, if you see a smaller fish that might be whirling or have a black tail, or maybe you can see its gills, it really helps biologists to report that information. And so here we have listed on the presentation a number that you can call, or you can send an email to aep.whirl@gov.ab.ca to report wild situations where you think you observe this. And the information we'd like to include is a location of where you saw possibly whirling disease, at what time, and what did you see, if you can describe it as best you can. And what this does is it helps inform biologists of where we need to be looking, because as we all know, Alberta is a very big place and we can't get everywhere at once. So having people that are concerned about whirling disease and actively looking for it really helps us direct our future work. And that's all I have about whirling disease. One other main point, just as I finish here, is now that you've learned a little bit more about it, I would encourage you to tell your friends and your family about it, because the more people that know about it, the more people are going to take those good behaviors of cleaning their gear, and the more likely we are going to be to prevent it from getting into places where it isn't, and therefore harder to deal with. So I would encourage that you do spread some of this information. So spread the information, not the disease. So that's all I have, and I'll pass it over to Robin to introduce Mad Lib. So thank you very much for everyone's attention today.
Thanks, Clayton, and that's great advice to spread information, not the disease. I'm sure that goes for COVID right now as well, hopefully. But to encourage you to tell your friends and family about whirling disease and maybe put a slightly more hilarious spin on something that's not so funny, we have created a whirling disease Mad Lib here. So if you're not familiar with Mad Libs, what you can do is the blue blanks there tell you what kind of word you need to think of to fill them in. And then you can read through this Mad Lib just like a story, and some of them get quite hilarious, but there's some great factual information in there as well to really share all about whirling disease. So after today's webinar, this will be posted on Bow Habitat Station's Facebook page if you wanted to go check that out and download it onto your device or your computer, and then you can work through this with family and friends in the coming days. And if you do have a really great Mad Lib that you'd like to share, we just encourage you to share it on social media with the hashtags whirling disease, clean drain dry, and bow habitat, which are listed right at the top there. Excellent. So with that, we will get into some questions here. So if you have any questions that Clayton didn't cover in this presentation so far, feel free to use the Q&A function or Q&A box and the chat box as well to ask those now. But we do have a few that came in already, Clayton. So one of them is, do fish have to be deceased in order to release the spores of whirling disease?
Oh, that's a very, very good question. Generally, that's how they're most often released, is from when they die and they rest on the bottom and they begin to decay. And actually the worms will come and eat the decaying fish, and that's how the likelihood of getting the spores increases. But there is evidence, there is some scientific study that have shown that spores can shake loose from the fish, and particularly the gill area. So there's a lot of cartilage in the gill, so when the spores form in that area, if they shake it out enough, they can release the spores. So there has been some evidence to show that spores can come out of live fish. In addition, if a fish gets eaten by another fish, or maybe a bird that eats fish, say like a great blue heron, the fish, once it passes through the stomach and the digestive system, the spores will still be intact. So if that ends up back in the river, it will still be viable and still can pass through. So that's a great question.
Another question that came in is, I know you said that whirling disease has two hosts and that one is a worm. Can other worms that live in the dirt transmit the virus, such as earthworms?
Another really good question. There's been a lot of work on this, actually. Other worms will eat the parasite. So we have, in our own studies, we have seen that other worm species are positive for the parasite. But it's only this very specific worm. In fact, in the literature, and other studies have shown that that worm has what are called different lineages. So it's called Tubifex. Tubifex is the name of the worm, and there are 5 lineages of Tubifex. So it's the same species, but just slightly different in their susceptibility to whirling disease. And it actually is only one of those 5 lineages that is susceptible. So the host is the parasite is very specific to that Tubifex worm.
Okay, great. And then can eDNA be used to test for parasite concentration?
eDNA can be used to test for the parasite. That has been shown to work, yeah. We actually send DNA samples. We send full worms, or even sometimes sediment like mud samples, to test for the parasite. But that's actually looking for the parasite DNA. But yes, there has been studies that show that eDNA can work for determining if the parasite is present.
And Clayton, for those of us on the line who are not familiar with the term eDNA, can you just tell us what that means?
Yeah, it's called environmental DNA. So how to describe this? There's DNA, like for human beings, if you were to look at our DNA, there's DNA coded in all of our tissue and all throughout us. But sometimes things shake off, like hair or skin cells and stuff like that. So that becomes environmental DNA. So DNA is now present in the environment. And so what eDNA is you take a sample, let's say of river water, and you filter it through a very fine filter. Some of that floating DNA will get captured. Particularly with this parasite, you may just be capturing those little TAMs, like part of the TAM that's flowing through the water, and that would be what eDNA, environmental DNA, would be.
Excellent. Thanks, Clayton. It looks like we have one more question that is about a fish parasite, but unrelated to whirling disease. Clayton, maybe I'll ask it, and if you're not the right person to respond to this, then we can follow up with this participant at a later date. So I recently got my dream aquarium fish, a giant freshwater MBU puffer. I recently corresponded with an exotic vet, and they said that I run the risk of disease or infection if I feed my puffer ram shorn and pond snails, like bladder snails and crayfish. Could you confirm this? Would freezing prevent this? And then they go on to say, thanks, you've been fantastic. Only answer if you have the time. I just appreciate your expertise.
Yeah, yeah, thank you so much. But in terms of the question, it is a bit specific to aquarium disease, which I'm not particularly familiar with. It sounds like a very, very specific situation. But I do think I know who I could definitely get somebody to touch base with Chelsea after. They might know how to answer that question better.
Excellent. So Chelsea, we will follow up with you. I do have your contact info, so stay tuned for that. Perfect. So with that, I think that is all of the questions that have come in. So as you ruminate on this presentation, if anything else comes up, please don't hesitate to email Bow Habitat Station, and that link will be coming, or our email contact will be coming out in an email following this webinar, where we'll also put the link to that AHIA fisheries education course, as well as the phone number for Clean Drain Dry. So stay tuned for that, and don't hesitate to ask us any questions. Oh, just as we wrap up, we had one more question coming in that said, cleaning your gear at home, are we not just washing the parasite downstream? Is it killed by the treatment plants?
That's a good question. Generally, you would clean your gear if you could rinse it off at the shore as you're leaving. That helps. But yeah, definitely clean it at home. That's very. I would. So what I do for myself is I'll go out into the front yard and I'll wash it on the front yard, and then I'll hang up whatever I've used to dry in the sun if it's possible. I don't believe it can go through the treatment plant, and it really depends on where you're located. For example, Edmonton, everything downstream of Edmonton, there aren't any susceptible species to whirling disease, so it's less of a concern. But to answer that question, it's very challenging to know if the parasite can make it through the treatment plant, but I would suspect not very likely.
Okay.
Yeah, sorry, it's a bit confusing. That hasn't really been studied that well.
Well, as you said, it's quite new, so I'm sure that there is lots more research to do in the future. We do have one message here saying the wastewater treatment plant has been experimenting with UV. I believe this is here in Calgary, so that could essentially kill that spore as well. So
absolutely, UV is a good way to kill the spore.
Excellent. Well, with that, I think we'll close for the day. So thank you so much to everyone who joined us, and thank you so much, Clayton, for coming on and sharing your expertise with us. I think I speak for everyone when I say we learned quite a bit from you this morning.
Awesome. Yeah, thanks again, Robin. Thanks for hosting this.
Yeah, anytime. So thanks, everybody, and we hope to see you at a future webinar. And I will just plug Creekfest here. So this year's event is running through to the end of day tomorrow. So if you check out, if you Google Creekfest 2020, there's some other great events that are happening throughout the rest of today and tomorrow. So I highly encourage you to go look into those, and yeah, enjoy. Perfect. Thanks, everybody. We'll talk to you soon. Bye.